Well, first off, I just want to say that I'm deeply honored for receiving the Herb Tabor Award tonight. And I would really like to thank my close friend and colleague Kim Worth for having nominated me, and for the selection committee for having selected me. As Kim has foreshadowed, this is a particularly heartfelt award for me to have won the Tabor Award, having worked in the polyamine field for my entire career. And Herb and his wife Celia pioneered the early discoveries on polyamine metabolism and were an inspiration to all of us in the field of polyamine research, but also in the field of biochemistry and enzymology altogether, based on the incredible discoveries that they made. So let's see, I'm going to be able to move forward. So I just wanted to just say, the seminal discoveries of Herb and Celia really parallel the discoveries in biochemistry as they came through from metabolites all the way to DNA. So their early work, they discovered the precursors that were needed to form polyamines. They then went on working in yeast and bacteria to discover the enzymes that catalyze the reactions to produce these polyamines, and then finally went on to identify the gene products that encoded these enzymes. And so the canonical polyamine pathway that Herb and Celia uncovered is really the pathway that's used by almost every cell to make these really important basic metabolites in the cell. And my interest in these came from my interest in working on these parasitic trypanosomatids, and from the hopes that I might be able to study polyamine metabolism to identify potential new drug targets. But what I found out along the way was that I uncovered a lot of really interesting biochemistry, because these ancient organisms had developed different approaches to some of the regulation and some of the ways they built their enzymes. And so tonight, to honor Herb, I decided to talk on my polyamines work as opposed to the work on malaria. I thought it was most appropriate for this particular award. So just to start with, to introduce these parasitic trypanosomes to you, there are 3 species that cause human disease: Trypanosoma brucei, Trypanosoma cruzi, and Leishmania species. And these are single-cell eukaryotes that many of you probably don't think about a lot. But what's interesting is they're from the very earliest branch point, one of the very earliest lineages off the eukaryotic tree, the euglenozoa, branching off, as you can see from this slide. I'm trying to see if this pointer works, but I'm not sure about the pointer. Okay, anyway, so I took this tree of life from Mark Field's review, and you can just see that the euglenozoa branch off a very early lineage off the eukaryotic tree. So these parasites have had a long time to evolve different types of processes than a lot of their eukaryotic cousins. So just a few words on the diseases that are caused by these parasites. All 3 of them share in common that they're transmitted by the bite of blood-sucking insects. And then they differ in terms of where they infect in the body and the type of disease they cause, which is really related to where these infections occur. Trypanosoma brucei is an entirely extracellular pathogen that infects the blood and the central nervous system, leading to neurological symptoms, whereas Trypanosoma cruzi and Leishmaniasis are intracellular parasites targeting either heart cells in the case of T. cruzi, leading to cardiac disorders, or intracellular parasites, Leishmania, invade the macrophages and then targeting the spleen and the skin in those cases. And in all cases, all 3 of these parasites cause fatal disease. And in all cases, the drugs that we have to treat these parasites are significantly inadequate. And that's really true, particularly for Trypanosoma cruzi, where really we don't have good drug therapies for the late-stage disease at all. So from this background, I really started my work in Trypanosoma brucei, trying to understand if we could target this polyamine pathway to develop new drugs. And so what I'm showing you here is the canonical pathway that's found in in most cells, and certainly in all eukaryotic cells. And that is that spermidine, the polyamine spermidine, is synthesized as demonstrated by Herb and Celia from the decarboxylated adomet and putrescine as precursors. And these two precursors are in turn synthesized by two key decarboxylases, ornithine decarboxylase, which generates putrescine, and S-adenosylmethionine decarboxylase, which generates decarboxylated Atomet. And then the N-propyl group from decarboxylated Atomet is linked onto putrescine by spermidine synthase, generating spermidine. Now, spermidine is a very important molecule to eukaryotic cells because it's essential substrate for the formation of deoxyhypozine modification of a translation factor called EIF5A. And both this translation factor and this modification are essential for eukaryotic cells. Now, these polyamines by nature are great chemical tools to synthesize other molecules. And in mammalian cells and most higher eukaryotes, spermidine then goes on to be elongated by an additional N-propyl group to form spermine. But in trypanosomes, it was discovered by Alan Fairlamb that instead of forming spermine, spermidine is conjugated to glutathione to form a redox cofactor called trypanothione, which supplants glutathione in these cells, and all the enzymology and chemistry that it's undergone in these cells to maintain redox balance now is focused on trypanothione. So that was the first of the very novel discoveries in this pathway in trypanosomes. We know from work in my lab and from work in Alan's lab that all the genes in this pathway are essential to Trypanosoma brucei by genetic knockdowns. And furthermore, a very important discovery by Saibaki in 1980 was that a suicide inhibitor called eflornithine, which targets ornithine decarboxylase, had in vivo anti-trypanosomal activity, and it later went on in 1990 to be approved for clinical use for the treatment of human African trypanosomiasis. This led us to really want to explore other enzymes in this pathway to see if we might identify other possible targets for additional drug discovery. And that led us to our studies on S-adenosylmethionine decarboxylase and deoxyhypozine synthase. There are a number of known inhibitors to ADO-MET-DC. I've just shown you one there. None have yet actually ever translated to the clinic. So today I'm actually not going to focus on the novel parts of this pathway, but actually on the canonical pathway to tell you some stories that we learned along the way that really reflect on biochemistry and enzymology in these parasites that differs from their eukaryotic cousins. So this pathway, as I said, is highly conserved across eukaryotes. It's also very highly regulated in mammalian cells at the levels of transcription and translation, with spermidine being the key regulatory molecule. However, the trypanosomatids, as I'm going to show you, have really evolved a number of novel features. And a lot of this novel chemistry that's occurring, it's really centered around functional divergence, gene duplication, and the role of pseudoenzymes in this pathway. And I'm going to show you stories where we've uncovered novel enzyme configurations involving pseudoenzyme paralogs of genes in the pathway, also novel regulatory mechanisms, and then in a final case, pseudoenzymes that have evolved other functions coming from this pathway. So as I said, I'm going to focus most of my discussion on Trypanosoma brucei as a model, but occasionally a lot of what I have to say will translate actually to the other trypanosomatid parasites, because what we found is true for the others as well. Okay, so starting out with the story on S-adenosylmethionine decarboxylase, this is an essential pyruvyl-dependent enzyme. So this is one of these enzymes that undergoes an autocatalytic cleavage reaction to generate a pyruvate, which is used in enzyme catalysis, and that pyruvate is at the N-terminus of what's termed the alpha subunit. So it was the larger one that was first seen on gels. And so in Trypanosoma brucei, I told you that this is an essential enzyme, and we could show this using genetic tools. We have a full range of those in Trypanosoma brucei. And in this experiment, I'm showing you where we induce the RNAi response by expressing a double-stranded RNA under the control of the TET promoter. And what we can see here in the middle portion of the slide is that this knockdown leads to a loss of ADO-MET-DC protein at the western level, and it also then leads to a growth arrest and followed by death of these parasites. We can rescue this knockdown by adding spermidine back into the cells. And then furthermore, we can show that spermidine is depleted upon this knockdown, really showing that ADO-MET-DC is essential for this formation of spermidine. But a surprise that we had when we looked at the recombinant enzyme and measured the steady-state kinetic parameters of this enzyme was that our recombinant T. brucei ADOMET-DC was over 1,000-fold less active than the human counterpart. And this really had us scratching our heads because most homologous enzymes tend to catalyze reactions with similar kcats. And so what we realized, though, was that there was a second paralog of ADOMET-DC encoded in all the trypanosomatid parasites, and that this paralog was actually had evolved further away from the canonical ADOMET-DCs. And so you see in the upper part the T. brucei, cruzi, and L major ADOMET-DCs that are more similar to the mammalian and yeast enzymes, whereas this one we're calling Prozyme, it has diverged further. It's missing catalytic residues, I'll tell you that now. It's found only in the trypanosomatid lineage. And we named it Prozyme for catalytically dead enzyme activator, which is already foreshadowing what I'm going to tell you it actually does. When my student Erin purified these two separately from E. coli, she found that, as I said, the ADOMET-DC was not very active. This Prozyme was completely dead, and as you see from this gel, it's not processed to form the two subunits, so it can't undergo the autocatalytic cleavage. It's missing the key serine residues. But if we mix them together or co-purified them, they form a complex that would co-purify over multiple columns. And so we could also show that if we flag tagged the ADOMET-DC in parasites and then pulled it down and looked by mass spec, that we'd pull down equivalent amounts of Prozyme, so they were clearly forming a complex. And then finally, when Erin assayed them, she saw that the ADOMET-DC Prozyme complex now was fully active as the human enzyme. And so it turned out that this ADOMET-DC required heterodimerization with this catalytically dead pseudoenzyme paralog to form the fully functional enzyme. And so another question we asked at the time was this just sort of regulating enzyme activity, or did this regulate the cell, the polyamine metabolism in the cells as a different mechanism from what mammalian cells use? And what we noticed was that whenever we knocked down ADOMET-DC, or we inhibited it with an inhibitor, as I'm showing here, a suicide inhibitor called Gen 64441, we would get induction of Prozyme protein expression. And in this case, after 6 hours of treatment, about 4-fold, though in knockdown studies we would see up to a 20-fold induction. What we could go on to show was that the level of regulation occurred at the level of translation and not at the level of RNA, and we could show that the rate of translation was actually increased in the presence of this compound Gen 64441. We could show that there was no correlation to spermidine, which is the key regulatory molecule in other eukaryotic cells, as if we knock down ornithine decarboxylase or spermidine synthase, we get no upregulation of this Prozyme. However, we found that we could replace the trypanosome enzyme with the human enzyme, and what we got was constitutive activation of Prozyme translation, and so that we had high levels of Prozyme all the time. So it appeared that the enzyme itself was actually involved in the regulation. And then finally, what we really noticed is an inverse correlation between Prozyme expression and the concentration of the product in the cell. So the less DC ADOMET we have, the more Prozyme we have. And to kind of try to provide further evidence that this molecule was involved, we knocked out ADOMET-DC synthase, and in this case, we also induced Prozyme expression. So it appears that this Prozyme not only is essential for activating ADOMET-DC at the enzyme level, but is probably playing a regulatory role in polyamine homeostasis in these parasites, and that's really a completely different model than what we see in mammalian cells. So turning then to the structural basis of the activation, and this is one of the coolest studies I ever did. I love the structural work that came out of this. My student Nair first noticed that the trypanosomes shown in this green box had an N-terminal sequence that wasn't found in any of the other eukaryotic enzymes, and that it was absolutely invariant in all 3 of these trypanosomatid species. And what she noticed, though, was that if she cut off the first 16 amino acids from the N-terminus, that this truncated delta 16 enzyme was no longer activatable by Prozyme and remained catalytically at low activity in any case. And we sent this work to JBC, and actually Herb Tabor was the editor, and he wrote my student, a very, very nice note when this paper came back from review, and I think that she and I both cherish that till this time. My postdoc Oleg then went on to solve the X-ray structures of this catalytically incompetent version of delta 16, and then he compared that to the active heterodimer. And what we immediately saw when we solved the structure of this inactive delta 16 mutant was that the N-terminus, I'm showing here the T. brucei enzyme is in pink and green and tan for the two subunits, with pink being the N-terminus and tan being with the pyruvyl-dependent side of the alpha subunit. And then green, it's aligned to the human enzyme. And what you see is that the N-terminus in pink has turned into the active site and is actually overlapping that small little inhibitor, green inhibitor, which is coming from the human enzyme. And so we have an auto-inhibitory residues actually parked in the active site. And there's a cis-proline that's conserved only in the trypanosomatids that allow this turn into the active site. And you see the human enzyme in green just that moves out in a different direction and is not impacting the catalytic site. And so then Oleg solved the heterodimer structure of the atom at DC bound to prozyme shown in gray here. And you see that the N-terminus is now flipped out and is bound in the dimer interface. And the active site is now open, and this inhibitor CGB4-2015 is bound in the active site of this now fully active enzyme. And interestingly, so the N-terminus docks into this interface, and at the same time that you get this flipping, you get a reorganization of the central beta sheet that basically builds the pocket for this N-terminus to flip into. So it was a really cool allosteric mechanism for how this inactive prozyme was activating the enzyme. Okay, so I wanted to turn now to the second story that we uncovered, because amazingly, there's not only one of these pseudoenzyme stories in this pathway in trypanosomes, but two. And so it turns out when we looked at the trypanosome deoxyhypocene synthase, which I told you is essential to make this hypocene cofactor on EIF5A, we found that, again, there were two paralogs encoded in the genome. One which we call DHSC, which contains the known catalytic lysine for deoxyhypocene synthase, but a second which we call DSHP, which did not have the lysine. And so it looked like, again, we were going to have another situation where we might require two partners to actually make an active enzyme. And that's indeed what happened. We look at the human enzyme-specific activity in gray here. The DHSC, the catalytic one, has a little bit of activity. The DHSP is completely inactive, but when we co-express them and purify them, we now get a fully functional and active enzyme. So again, it appears that both of these are required for activity. In this case, we have to co-express the two together, and you can't just mix them together. They're forming a much more tight complex. Both are essential when we do genetic studies, but in this case, if you knock one down, the other disappears too, just indicating how reliant they are on each other. Now what I'm going to show you is that the mechanism of activation in this case is at the structural level is actually completely different from that first story I showed you. So in this case, this enzyme in the human case there in gray is a tetramer, but the active unit is really a dimer, and the active sites are formed at the dimer interface, and there's two active sites per dimer. Where they bind NAD and then the spermidine as two substrates, and then the EIF5A would come in as the third substrate. But what we noticed really immediately upon looking at the sequences is that the DHSC component really had amino acid substitutions in the NAD binding site so that you wouldn't predict that it would be as active, whereas the DHSP protein had mutated residues in the spermidine binding site, suggesting it wouldn't be active. And so this suggested to us that we were going to get a shared active site structure, forming one functional active site that I've shown with the green circles, and then one double dead active site that I've shown with the tan circles. So the prediction is then that the T. brucei enzyme would have two functional active sites and two double dead sites, and that really, in this case, the two pseudoenzymes now can only form an active enzyme by actually binding to each other. And primarily, this analysis would suggest the NAD binding site would be on DHSP, mostly in the spermidine site, including the catalytic lysine on DHSC. So we solved a number of structures, initially a 3.5 angstroms crystal structure, but recently I can tell you we've just solved a 2.5 angstrom cryo-EM structure bound to NAD and to a molecule called GC7, which is a spermidine analog inhibitor of the protein. and also a cryo-EM structure, which we thought was going to be the empty structure, but as I'll show you, contains 2-NAD anyway. So as expected, if we look across, this is the tetramer, and there's a dimer interface at the top and the bottom. And you can see that I've got 4 NADs bound. The green ones are the active site, and the tan ones are in the inactive site. And so the initial surprise was that we actually have NAD bound in the inactive site. And that hadn't been expected based on the amino acid substitutions that we had seen. However, the GC7 is bound to only 2 sites, and it's bound only to the active sites packed up against that active NAD. So if we look a little closer now at the details, I'm looking now at just one dimer interface. The green, again, is the active site, and the tan is the inactive site. And you see the GC7 then packed up against the active NAD. And we can make a couple observations. The NAD in the dead site is actually in a non-productive conformation where the nicotinamide head has been turned. And if we make mutations in the dead site, they don't impact the catalytic rate, though they will change the Km for NAD binding. The cryo-EM structure, the third one I told you about, where we had thought it was going to be unliganded because we gave it no ligands, but in turn, what we saw was it was actually still bound to 2-NAD. Those NAD are bound in the dead site, suggesting that NAD is bound tightly to the dead site and playing a structural role. And that makes sense because the NADs are actually within van der Waal contact of each other. This would also be true of the human enzyme when both sites are active, but in our case, I think that you had to maintain this binding of NAD to actually form the site. And then if you look at the GC7 site, again, you can understand why the dead site is dead. It's actually replaced a number of the residues that are key with residues that actually fill in and obstruct the dead site binding for spermidine or GC7. So in this case, it's quite clear, again, the structural basis here is a shared active site structure. Okay, I just wanted to, in the final minutes, tell you one final short story. So I've told you now that the trypanosomatid parasites have used inactive pseudoenzymes to form the active enzyme structures of 2 enzymes in the pathway, S-adenosylmethionine decarboxylase and deoxyhypocene synthase. In the Adamet DC case, they also seem to have developed a novel regulatory mechanism. But we've also uncovered evidence that there are pseudoenzymes in other parts of the pathway that now play different functional roles altogether or are completely catalytically dead. So we turned to a recent student of mine, Michael Panikos, wanted to ask the question of how the parasites actually generated ornithine to form putrescine. And so it turns out, while Leishmania donovani, one of the 3 parasitic trypanosomatids, has an active arginase, the arginase in T. brucei is catalytically inactive, again, apparent pseudoenzyme. But in this case, there doesn't seem to be any other paralog or any other way that it would get reactivated. Ornithine transporters have been reported in T. brucei, and so the hypothesis is potentially that that T. brucei anyway was an ornithine auxotroph. But the levels of ornithine in spinal fluid are actually quite low, so we are a little perplexed by that, and we thought, well, maybe there's another enzyme that can produce ornithine in these parasites. So we identified an amidotransferase in these parasites, and this enzyme takes arginine, transfers the amido group to other amine donors, and then generates ornithine in the process. And so we thought, well, maybe this enzyme is capable of generating ornithine in these trypanosomatid parasites. So we decided to study this enzyme a little bit further. And so Michael in my lab expressed this enzyme, which I'm just calling TB927-1300, in E. coli, and he found that it had no activity versus a range of acceptor amino acids or substrates that we could potentially know about from the literature and other amidotransferases. We had a bacterial control enzyme called HSVA, which is a polyamine-dependent amidotransferase, which showed robust activity in these assays. So we certainly were able to assay the enzyme. And there didn't appear to also be another paralog of this enzyme in T. brucei or in any of the trypanosomatids that might, recapitulate its activity as in the earlier cases. So we did one further experiment. We incubated the recombinant enzymes in T. brucei lysate to see if maybe there was another small molecule metabolite. We were missing the substrate, something like that. And what Michael did was he then analyzed after these incubations by untargeted mass spec analysis or by targeted mass spec analysis to look to see the change in key metabolites in the cell. And so again, we saw no activity for this T. brucei gene product, 927-1300. There's really no change in any of the relevant metabolites. But in the case of this bacterial enzyme, we saw, going backwards here. We saw robust activity in the way that we would expect. Arginine decreased, ornithine and agnitine increased, and then additionally we identified an unexpected metabolite called 3-aminopropyl-agnitine, which told us that the spermidine synthase in T. brucei must be fairly promiscuous and able to work on arginine. So that really pretty much told us that this protein we found, let's not call it an enzyme, was not likely to be catalytically active. And we finally then looked through isotype tracer studies using C13 labeled arginine, and this was not converted by T. brucei to ornithine, leading us to conclude it's likely an oxytroph. But from the point of view of our pseudoenzyme talk today, it seems that this is actually an inactive pseudoenzyme that's been co-opted for new function, as Ian Lee has recently reported it to be associated with microtubule cortical cytoskeleton, and both he and we found that it is essential to the parasites, and there is a growth effect when you knock it out. And so just to conclude then, what I've told you today is that in the trypanosomatids, they've taken this highly conserved eukaryotic pathway that is absolutely essential for all eukaryotes because of the role of spermidine in activating EIF5A. And we've identified two enzymes in the pathway that rely on inactive pseudoenzymes to generate the active enzymes. In one case, ADAMET-DC, the pseudoenzyme, which we've termed prozyme, actually appears to play a regulatory role in polyamine biosynthesis. And then we've identified a couple of other pseudoenzymes where their function has been, they're no longer enzymatic but appear to play other structural roles in the cell, or in the case of the arginase, actually we don't yet know what it might be doing in the cells. So with that, I'd just like to thank all the people in my lab who've done this work over the years. Aaron Willert and Oleg Volkov really pioneered the work on ADAMET-DC. In the deoxyhypusine synthase work, it was pioneered by Sun Nugent in my lab. It was a collaboration with Alan Fairlam. Gustavo Afandider and Xiaoyi Deng have done all the structural work. And the aminotransferase work was recently done by Michael Panikos in the lab. I'd like to also thank the UT Southwestern Structural Biology Corps, who was really a key resource and phenomenal help in all of this. And also my husband, Tony Michael, who I talked to a lot, especially about the evolutionary aspects of polyamine biosynthesis. So with that, I'd like to thank you for your attention, and happy to take questions.